Greetings. We are first year biomedical science students presenting a video on the differentiation between Staphylococcus epidermidis and Staphylococcus aureus as a part of our e-portfolio assessment. To begin with, we will explain what Staphylococcus epidermidis is and Staphylococcus aureus. Staphylococcus epidermidis is a gram-positive bacteria which forms clusters on microscopic examination. It is a normal inhabitant of human skin and also mucous membrane, and also considered as less pathogenic than Staphylococcus aureus. Staphylococcus aureus is a gram-positive bacteria which forms clusters resembling grapes on microscopic examination. It is a major opportunistic pathogen which found on the skin and also mucous membrane of the human. Specimen type of S. epidermidis are wound swabs, skin swabs, blood, urine, and respiratory secretions. Meanwhile, the specimen type of S. aureus are blood, urine, wound swabs, and catheter or implants. Patients with S. epidermidis infection, they usually present with low blood pressure or rapid heart rate in severe cases. In addition, they also have fever and chills, especially in bloodstream infections. Patients with S. aureus infections, the general symptoms are fever, fatigue, and chills. And if it's a skin infection, they may experience pain, swelling, and redness.
For the stepwise identification of Staphylococcus species, the first approach involves microscopy and staining. This step allows us to observe the morphology, arrangement, and gram reaction of the bacteria. By using microscopic examination and gram staining, we can narrow down the organism to a specific bacterial genus, which is an important foundation before proceeding to further biochemical tests. However, microscopy and staining have their limitations. These methods cannot differentiate between Staphylococcus aureus and Staphylococcus epidermidis. This is because both species show the same characteristics, which are gram-positive and purple stain cocci arranged in clusters. As a result, microscopy alone is insufficient for species-level identification. Therefore, further biochemical tests are required to accurately differentiate between the Staphylococcus species.
For culture characteristics in differentiate between Staphylococcus epidermidis and Staphylococcus aureus, we can use two different culture media to differentiate both of them, which is blood agar and mannitosol agar. For the Staphylococcus epidermidis, the blood agar will show gamma hemolysis, which is non-hemolytic. This is because there is no production of hemolysins, so the red blood cell will remain intact. The colonies will also appear small medium with white or grey color. In mannitosol agar, the Staphylococcus epidermidis will show a growth with red medium. This is due to the negative mannitofermentation, which do not produce acid and remain the color of the medium. For Staphylococcus aureus, the blood agar will show a beta hemolysis with a clear zone around the colonies. This is due to the production of hemolysins that completely less the red blood cell. The colonies also appear mediums, smooth and golden. In mannitosol agars, the Staphylococcus aureus will show a growth with yellow medium. It can tolerate with hard salts and has positive mannitofermentation that produce acid, which then turns the phenol red color into yellow color.
Next, we are going to look at the mannitofermentation test. The purposes for this test is to determine the bacteria growth by encouraging certain bacteria growth and inhibiting other bacteria. The method for this test is by inoculating the organism into mannitosol agar, then incubate at 37 degrees Celsius for 34 hours before we observe the color changes of mannitosol agar. For the result, the Staphylococcus aureus show a positive result as the medium turns into yellow because it lyses the red blood cell, while the Staphylococcus epidermidis show negative result as the medium stay red or pink color, showing no reaction occurred.
Today I'm here to explain about the biochemical and antigen-based tests for the Staphylococcus aureus and also Staphylococcus epidermidis. So there are three biochemical and antigen-based tests. So today I'm here to explain about the Coagulase test and also lattice aggregation test. So Coagulase test. So first we see the purpose of the Coagulase test. So the purpose of the Coagulase test is to distinguish the Coagulase positive from the Coagulase negative staphylococci. So the methods that we use to to perform the Coagulase test is a slide or a tube Coagulase test using the plasma. So the result and the interpretation that we got from the both Staphylococcus aureus and also Staphylococcus epidermidis are a positive Coagulase tested strongly indicated Staphylococcus aureus, and a negative result suggests Coagulase negative staphylococci such as Staphylococcus epidermidis. So the second biochemical test and antigen-based test are lactase agglutination test. So first we see the purpose of the lactase agglutination test. So it is a rapid detection of protein A and clumping factor on the bacterial surface. So the methods that we use to perform lactase agglutination test are: a bacterial colony is mixed with latex particle coated with specific antibodies on the site, and a visible clumping indicates a positive result. So from here, if the test shows a visible clumping, so it is positive towards lactase agglutination test. So the result that we got from the lactase agglutination test are: Staphylococcus aureus shows a positive agglutination where there is a visible clumping, and also Staphylococcus epidermidis shows no agglutination, which means there is no visible clumping. So what we can interpret from the both of the results is a positive lactase agglutination test confirms the presence of Staphylococcus aureus antigens, providing a rapid diagnostic confirmation.
To differentiate Staphylococcus aureus and Staphylococcus epidermidis, molecular and rapid diagnostic methods such as PCR, multiplex PCR, and the latex agglutination tests are commonly used because both organisms appear very similar under the microscope. Firstly, polymerase chain reaction PCR is a highly specific molecular method that detects bacterial DNA. In this technique, species-specific genes are amplified to identify the organism. The nuke gene is a well-established marker for Staphylococcus aureus and is not found in Staphylococcus epidermidis. On the other hand, Staphylococcus epidermidis can be identified by the presence of genes such as ICA gene, which is associated with biofilm formation and is commonly found in coagulase negative staphylococci. PCR provides high sensitivity and specificity, allowing accurate differentiation between these two species within a few hours. Secondly, multiplex PCR is an advanced form of PCR that allows the amplification of multiple target genes in a single reaction. This method can simultaneously detect species-specific genes such as nuke gene for Staphylococcus aureus and ICA gene for Staphylococcus epidermidis. By combining several targets in one test, multiplex PCR reduces time, cost, and sample volume while providing comprehensive diagnostic information, making it especially useful in clinical and hospital laboratory settings. Finally, the latex agglutination test is a rapid non-molecular screening method used for quick differentiation. This test detects protein A and clumping factor, which are present in Staphylococcus aureus. A positive agglutination reaction indicates Staphylococcus aureus, while Staphylococcus epidermidis, which lacks these factors, gives a negative result. Although this test is less sensitive than PCR, it is simple, fast, and useful for preliminary identification before confirmatory molecular testing. In conclusion, PCR and multiplex PCR provide accurate and reliable differentiation at the genetic level, while the latex agglutination test offers rapid initial screening in routine microbiology laboratories.
Next is spider identification and justification for two bacterial cases. For case 1, the organism was identified as Staphylococcus epidermidis. The gram stain showed gram-positive cocci arranged in clusters. On blood agar, the colonies were white and non-hemolytic, which there was no breakdown of red blood cells. On MSA, the organism showed no mannitol fermentation. The bacteria was catalase positive but coagulase negative. The latex agglutination test was also negative.
These results are consistent with Staphylococcus epidermidis. For case 2, the organism was identified as Staphylococcus aureus. The gram stain also showed gram-positive cocci in cluster. However, on blood agar, the colonies were golden and beta-hemolytic. On MSA, the organism fermented mannitol, causing a color change. The bacteria was catalase positive and coagulase positive. The latex agglutination test was positive. These key features confirm the organism as Staphylococcus aureus. In conclusion, the laboratory results clearly differentiate two organisms and support the final identification for both cases.